So who cares? Why do we care about this? Well, it turns out I need to do a little bit of a tutorial on protein shape. We talk about protein so much during the course of this class. Hopefully, this is one of our very last lessons, so hopefully you have started to internalize the importance of proteins and the fact that their shape is extremely important to their function. So, let's give a demonstration of this. This is one that you probably learned in general biology, the idea of protein, of the sodium potassium pump. The sodium potassium pump is a protein for which shape is crucial. The shape of proteins, in this case, allows you to have a binding region for the phosphate that comes from the ATP that activates the thing. And when the phosphate binds, it gives off this endergonic reaction, yes, so that it changes the configuration of the sodium potassium pump. It allows sodium ions to be pushed out of the cell. It allows potassium ions to be pushed into the cell, and both of them against their concentration gradient. And that is why it is called a pump, and that is why ATP is required. That is why energy is required to allow this to happen. So it's not your regular old protein that just kind of sits there like a channel. It is a pump. It is active. It does stuff. It pushes things. It makes things happen. The sodium potassium pump is incredibly important. We couldn't survive without it. We have to have that gradient. We have to have excess sodium ions outside of the cell. We have to have excess potassium ions inside of the cell, and the sodium potassium pump is the protein that allows that to happen. Okay, so we get the idea here. You need a particular shape. You need it to be embedded within the phospholipid membrane. You need it to have binding sites for potassium. You need it to have binding sites for sodium. You need it to have a binding site for phosphate. And so you get the idea. The shape of all of these things must be in place. Any slight deviation from appropriate shape, it's probably not survivable. This is what we would call one of those essential genes, the gene that allows for the sodium potassium pump to be transcribed and then translated and then embedded in the membrane. That gene is an essential gene. An essential gene, you can't survive without it being properly transcribed and translated. So where does the shape come from? So let's take a look at this. This is what we call this would be considered the secondary shape of a protein. This is an alpha helix here. And I like this picture because it shows the bonds that take place that actually allow for this alpha helical shape to be carried out. So an alpha helix or a beta sheet are the standard secondary shapes of proteins that they kind of just exist in all the time. And there are other shapes. There are tertiary shapes and quaternary shapes that may be forced by, for example, cysteines and disulfide bonds and specialized things within the protein, within the amino acid configuration itself. But in this case, if we just look at the basic amino acid, so the amino acid has a carboxyl end and it has an amine end and it has that central carbon and it has a variable group. We're not even worried about the variable groups right now because the variable groups will give the protein its special, probably tertiary structure or allow it to be embedded in a membrane or something like that. But in this case, we're only worried about the secondary structure. We're only worried about that alpha helix. So hopefully I'm not losing you with all of this because my hope is that you have had exposure to some of these concepts in the past, so it's not too tricky for you. So anyway, pay attention to this. At the moment, it's in an alpha helix, and the reason it's in an alpha helix is because you have hydrogens and oxygens that are bonded to each other. These are called hydrogen bonds. Here's the hydrogen of one amine group bonded to the carbon that looks actually like a central carbon. Here we have the hydrogen of one amine group bonded to the oxygen of a carboxyl group. Here we have a hydrogen here from the amine end of an amino acid bonded to the oxygen of a carboxyl group. So these are what we call hydrogen bonds. The central theme here is that you have a hydrogen that is loosely bound to some slightly negatively charged structure on the other end. So the hydrogen carries a slightly positive charge, the oxygen carries a slightly negative charge, and so on. Okay, so this then forces a kink in the structure. It allows for sort of a temporary bond that allows for these slightly positively charged things and slightly negatively charged things to be attracted to each other. The bond is not strong. The hydrogen bond is infamously weak, actually, and very tenuous, therefore. So the idea is that it could ostensibly break easily. So this is where pH comes in. Remember what pH is? pH is the idea that you need to have exactly the correct number of hydrogen ions in order for your biological processes to go forward. Here, we say that our blood is at a pH of about 7.4, and that means it has just fewer than 10 to the minus 7, the concentration of 10 to the minus 7, just below that number in order to balance out what's happening here. So what I want you to do is draw in your notebook like one circle that represents a very acidic substance, and I want you to draw a ton of hydrogen ions in there. I want you to draw another circle that is a very basic or very alkaline substance, which will have very, very few hydrogen ions in it, so that you can kind of picture in your mind the idea that acid equals hydrogen ions, and alkaline equals very few hydrogen ions. Either case is disastrous. It upsets the balance here. Remember, this secondary structure of a protein is mediated by these very tenuous, very weak hydrogen bonds. So let us say then that your first circle, where you have tons of hydrogen ions, is dumped adjacent to this particular protein. What do you suppose will happen? Now, I know that you're not going to be ambitious enough to draw this whole thing, but just picture in your mind that you dump a bunch of positively charged hydrogen ions into this, and now they're sort of mixing around this protein, this amino acid secondary structure. What do you suppose is going to happen? Is it possible that this negatively charged oxygen, instead of being attracted to this guy, might be attracted to the positively charged hydrogen ions that are swimming around now? And this guy, same story. This guy, instead of being attracted here, will be attracted to some of the hydrogen ions outside of here. This is a massive problem. And yes, indeed, that will happen. That is the nature of an acidic substance. That is why acids are so very dangerous, because they will cause these bonds to break. As I suggested, the hydrogen bond is already not a very strong bond. And so if you dump a bunch of acids, a bunch of positively charged structures in here, those oxygens are going to be disturbed from their regular hydrogen ion configurations, the bonds that they are supposed to be making, and they will denature. This then will denature the protein. So that's the importance of pH.